Hello students, today we will learn about microbial production of foods and beverages. Let's begin. It is estimated that about 20 to 30% of the household budget is spent towards foods in the developed countries. This may be a little less in the developing nations. Therefore, food and beverage biotechnology occupies a prominent place world over. There are records that man was making bread, wine, curd, etc. as early as 4000 BC before Christ. These processes collectively referred to as traditional or old biotechnology, most employed for the preparation of foods and beverages were based on the natural capabilities of the microorganisms. With the advance made in microbiology and recently biotechnology, food and beverage production is a major industry. Food biotechnology is also concerned with the improved quality, nutrition, consistency, color, safety, and preservation of foods, besides making them available around the year. In addition, modern biotechnological processes also take into account the health aspects of the people. Now, let's know about fermented foods. The production of fermented foods is variable. This depends on geographical region and availability of raw materials, traditions, and food habits of the people. Now, let's know about the advantages of fermented foods. It enhances nutritive value. Second is it increases digestibility. It improves flavor and texture. Fourth point is it serves as supplements in preparing several dishes. There are many other compounds that are directly or indirectly used as food supplements, which must also be referred by the reader. These include vitamins, amino acids, organic acids, enzymes, biopolymers, single cell proteins, and mushrooms. Now, first we will discuss about cheese. Cheese production is the largest dairy industry in the world. There are around 1000 types of different cheese. They are broadly two types: unripened cheeses and ripened cheese. Unripened cheese means cottage cheese with low fat, cream cheese with high fat. Cream cheese with high fat, and ripening cheese is hard cheese, that is example cheddar, blue cheese, soft cheese, example Limburger, camembert. Irrespective of type of cheese, all of them are invariably made from the casein of milk that is produced after separating the whey, that means liquid portion of the milk. Milk from different animals can be used, example sheep, cow, goat, buffalo. Now let's know about the historical perspective. The use of animal stomachs for carrying liquids is centuries old. When milk was transported in this fashion, the formation of solids that were tasty was observed. The solids were concentrated after draining liquids. These solids were salted and consumed later. A good example of the food preservation long ago, we now know that this solid portion is the cheese. It is produced by the combined action of enzymes, that is rennet of the stomach, living and bacterial contamination. Now let's know about the production process. Cheese is produced from milk. This is carried out by a process of dehydration wherein casein, that is milk protein, and fats are concentrated 5 to 15 fold. Cheese production is very complicated and broadly involves four stages: acidification of milk, coagulum formation, separation of curd from whey, and ripening of cheese. Now let's know about the acidification of milk. By employing lactic acid bacteria, that is Streptococcus lactis, Lactobacillus lactis, the sugar of the milk, that is lactose, can be converted to lactic acid. This lowers the pH to around 4.6 and thus acidifies milk. Now, let's know about coagulum formation. When the acidified milk is treated with rennet, that is the enzyme chymosin of animal or fungal origin, casein gets coagulated. Casein mainly consists of three components: insoluble alpha and beta casein and a K casein that keeps them in soluble state. By the action of chymosin, K casein is degraded. Consequently, alpha and beta caseins and the degraded products of K casein combine to form a coagulum, that is curd. This process of coagulation is dependent on calcium ions. Now let's know about separation of curd from whey. When the temperature of the coagulum is raised to around 40 degrees centigrade, the coagulum and whey gets separated. That means fluid portion is separated. The separated curd is cut into blocks, drained and pressed into different shapes. Now let's know about the fourth step, that is ripening of cheese. The flavor of raw cheese such as cheddar is bland. Ripening imparts flavors besides making changes in its texture. The procedures adopted for ripening are highly variable depending on the type of cheese to be prepared. The blocks of curd separated are subjected to the action of proteases, that is proteases and lipases. Alternatively, they may be inoculated with certain fungi, example Penicillium roqueforti. The hydrolysis of proteins and fats results in certain compounds which imparts flavor to the cheese. Mild hydrolysis of fats or cheese usually carried out by lipases or Aspergillus niger or Mucormyceae that results in butyric acid formation with characteristic flavors. Now, let's know about sources of chymosin for cheese production. There are several sources of rennet, that is chymosin enzyme for cheese production. These include calves, adult cows, pigs and fungal sources. Fortunately, the fungal, example Mucormyceae, sources of chymosin are almost comparable to the animal sources and are widely used in some countries. However, some people always prefer animal rennet used cheese due to its slight superior flavor. Now let's know about genetically engineered microorganisms for chymosin. Some workers could successfully clone the genes of animal chymosin and transfer the same into microorganisms. The chymosin so produced by genetic manipulation is very widely used these days in some countries. The taste of cheese manufactured by microbial chymosin, that is genetically engineered, and the animal chymosin are identical. Public as well as the vegetarian societies in some countries have adopted the cheese produced by the genetic manipulation of the enzyme chymosin. Now let's know about the yogurt. Yogurt is produced by fermenting whole milk by employing a mixed culture of Lactobacillus bulgaricus and Streptococcus thermophilus. While Lactobacillus bulgaricus, in short L. bulgaricus, produces acetaldehyde that imparts a characteristic taste, S. thermophilus results in the formation of lactic acid to give acid flavor. In addition, both these bacteria produce extracellular polymers that increase the viscosity of the fermented milk. Yogurt is very delicious and in fact, frozen yogurt is becoming popular as an alternative to ice cream. Now, let's know about sauerkraut. Sauerkraut is prepared in most Western countries. It is fermented and preserved from form of cabbage. The shredded cabbage is mixed with salt and packed anaerobically. High salt concentration promotes leakage of sugars from the cabbage while reducing the water activity. As the growth of the lactic acid bacteria occurs, the pH is lowered. At this low pH, the putrefying bacteria cannot grow. In this way, the sauerkraut can be preserved for long. Sauerkraut is nutritious as well as delicious. The traditional pickle, that may be mango or lemon, is in India, are also good examples of fermentation and preservation based on the principles of biotechnology. Now let's know about bread. Bread making from the dough by employing microorganisms is one of the oldest examples of fermentation process known to mankind. There is evidence that bread was prepared in Egypt in 3000 BC. Primarily, bread is a fermented product of cereal flours such as wheat and rye. The cereal flour mixed with water, salt, sugar and other ingredients is subjected to fermentation by yeast Saccharomyces cerevisiae, that is the top fermenting strain. The main reaction that occurs during bread formation is the fermentation of hexoses to carbon dioxide and ethanol. That is, the reaction is C6H12O6 gives rise to 2C2H5OH plus 2CO2. The ethanol produced either gets evaporated or forms esters. The carbon dioxide gets entrapped in the dough, resulting in its expansion. The expansion and stretching of the dough, particularly with wheat, is due to the unique elastic protein, namely gluten. Gluten is mainly responsible for retaining the shape of bread. Besides yeast enzymes, the enzymes example amylase of other microorganisms also help in fermentation and baking of bread. The texture of the bread is influenced by fats and emulsifiers added to the dough. The bread making is carried out with three objectives. First is good living due to good leavening due to carbon dioxide formation. Second is flavor development. Third is good texture. The yeast fermented bread has these characteristics. This is in contrast to the bread produced with baking powder, which also produces carbon dioxide. But this does not have the same flavor and texture as that produced by yeast. Thus, the yeast, which is appropriately referred to as baker's yeast, is a package of enzymes to give a desired product. In recent years, some workers have reported the development of genetically engineered strains of Saccharomyces cerevisiae with improved fermentation properties. Such organisms, when used in baking industries, are believed to further enhance the quality of bread with regard to flavor, texture, etc. Now, let's know about sourdough breads. In some parts of the world, sour bread are prepared by using the yeast Candida milleri and Bacterium lactobacillus San Francisco. Now, let's know about the baker's yeast. The living cells of aerobically grown Saccharomyces cerevisiae are collectively referred to as baker's yeast. Baker's yeast is commercially available either as a dried powder or dry yeast, available either dry yeast with about 95% dry weight or in the form of cakes, that is about 25 to 30% dry weight. These commercially available yeast preparations can be used in bread making. Now let's know about the production of baker's yeast. The medium for baker's yeast production contains molasses, ammonium salts or ammonia, vitamins, phosphates, and antifoam agents. Sugarcane or sugar beet molasses can be used. A commercially available molasses with a sugar concentration of 45 to 50% is usually preferred. Baker's yeast production is carried out by an aerobic feed batch process. The actual production process and the strain of Saccharomyces cerevisiae used depends on the company. The desired strain is maintained in a frozen state. The inoculum is prepared in in stages so that large volumes are finally obtained. From the inoculation reactor, that is fermenter, the culture is transferred to production reactors. The process is carried out in airlift or bubble column type reactors at pH 4 to 5. Temperature 28 to 30 degrees centigrade for about 12 to 18 hours. The yeast cells are washed, centrifuged and then dewatered on a rotating drum. The fresh yeast obtained can be either directly used or dried and stored. The yeast cells may be mixed with plasticizer, example vegetable oil, and prepared in a block form. This block can be cut into small pieces, wrapped and stored at 4 degrees centigrade until used. The samples of yeast are usually tested for baking properties before they are put in use. Now let's know about other vegetarian products. Besides the bread which is consumed worldwide, there are several other vegetarian fermented foods. These include rolls and cakes, soy sauce, idli, tempeh. That is, tempeh is used in Indonesia. So now let's know about coffee, tea, and cocoa. Coffee, tea, and cocoa are very popular non-alcoholic fermented beverages and extensively consumed throughout the world. When the tea leaves are crushed, the chemical components of tea are released by enzymatic activity. For coffee and cocoa, a natural fermentation process, that is by bacteria, yeast and fungi on the pulp surrounding beans, results in the development of flavor and aroma. The exact nature of the fermentation process involved in tea, coffee and cocoa are not fully known. The dried products, namely tea leaves and coffee and cocoa beans, are commercially available for beverage preparation. Now let's know about the sweeteners. Sweeteners occupy a predominant place in food consumption by all the people. The most predominantly used natural sweetener is sucrose. Sweeteners are invariably required for the preparation of soft drinks, ice creams, jams, jellies, sauce, bread, confectionery, pickles, etc. Hence, there is a continuous search for low-calorie sweeteners. Saccharin. Saccharin, a chemically synthesized sweetener, 30 times sweetener than sucrose, was used for the several years and its use is now being restricted due to certain adverse health effects. High fructose corn syrup HFCS produced from sucrose is sweeter, that is 1.5 times than sucrose. Aspartame, that is in aspartyl phenylalanine, is a product of innovative biotechnology. It is about 200 times sweeter than sucrose, and is truly a low calorie sweetener. Apart is apart this is improved for human consumption and is widely used in soft drink and other industries. Now let's know about thaumatin, a sweet protein. Thaumatin is a protein extracted from the berries of the Thaumatococcus danieli, a native plant in Africa. Thaumatin is about 3000 times sweeter than sucrose. Thaumatin certainly appears to be a good sugar substitute and a low calorie sweetener. It is in fact being used as a sweetener and flavor enhancer of various foods. At least 5 different forms of thaumatins with a molecular weight in the range of 20,000 to 22,000 have been isolated and characterized. Now let's know about genetic engineering of produce thaumatin. Genetic engineering to produce thaumatin. Natural production of thaumatin is very limited and therefore can never meet the demands of consumption. Biotechnological production of this sweetener is the method of choice for its large-scale manufacture. Thaumatin encoding mRNA species have been isolated, identified and converted to cDNA and finally a double-stranded DNA. These DNAs were cloned and transferred to E. coli, but the yield of thaumatin production was very low. Attempts were also made to express thaumatin genes in yeast such as S. cerevisiae and Clu- Clu- Clueveromyces lactis. Now let's know about monelin. Monelin, a candidate for sugar substitute. Monelin is a protein found in the fruit of an African plant named Discorophyllum cuminsi. It is about 1 lakh times sweeter than sucrose on molar basis. Monelin is a dimer composed of an A chain, that is 45 amino acids, and a B chain, that is 50 amino acids. This protein is optimally sweet when both the chains are held together and not when they are separated. It has been possible to chemically synthesize a gene that encodes both A and B chains as a single polypeptide. This gene was in fact introduced and expressed in tomatoes for the production of monelin. However, the sucrose has been very limited. Now let's know about flavor enhancers. Taste and flavor are very important for the acceptability of foods. Monosodium glutamate is the most commonly used flavor enhancer. Its production and other aspects are we can study next part. The degraded products of proteins and RNAs also impart flavor. Thaumatin and monelin are also flavor enhancing agents besides sweetening. The other flavoring agents are citric acid produced by Aspergillus niger, acetone and butanol produced by Clostridium acetobutylum. The lactones formed by some microbial fermentations also enhance flavor. Example: gamma butyrolactone produced in yeast fermentation of wine and beer. Mention must be Mention must be made about alcohol also, which can increase flavor and taste when added at appropriate concentrations. Example: certain medicinal syrups. Now let's know about alcoholic beverages. Alcoholic beverages are produced throughout the world. The type of the beverage and the substrate used for its production mostly depends on the crops grown in the region. For instance, in Poland, Russia, and Scandinavia, beers manufactured from barley are consumed, while in France, Italy, and Greece, wines obtained from Palmyra juice are mostly consumed by rural people. Now let's know about historical perspective. Arch- uh, Archaeological evidence indicates that there existed fermentation of grains as early as 4000 BC. Production of alcoholic beverages is considered to be a part of human civilization. The biological and chemical principles underlying alcoholic beverage production were elucidated by Louis Pasteur in the second half of 19th century. Now let's know about Saccharomyces cerevisiae, the key organism for the production of alcoholic beverages. The most commonly used organism for the production of beverages is the yeast, that is Saccharomyces cerevisiae. This organism is capable of utilizing simple sugars, that means glucose, fructose, and converting them to ethanol. It is fortunate that S. cerevisiae is a unique organism that can tolerate and grow even at a high concentration of alcohol. This is advantageous for good production yield of alcoholic beverages. There are two types of S. cerevisiae strains, bottom yeast and top yeast. The bottom yeast settles to the bottom, whereas the top yeast rises to the top while carrying out fermentation. Top yeast are frequently used for the production of beer, while bottom yeast are employed for wine production. However, both strains can be used for beverage production. Genetically engineered strains of S. cerevisiae. Let's know about genetically engineered strains of S. cerevisiae. By employing recombinant DNA technology, new strains of Saccharomyces cerevisiae are constantly being developed. Some of these strains with improved production yield of alcoholic beverages are in fact approved for use in beverage industries. Now let's know about alcoholic beverage production general aspects. The starting material for beverage production are sugary or starchy materials. Now let's know about sugary materials. That is fruit juice or fruits, plant sap and honey that can be directly used. Second is starchy material, various strains and roots. They have to be subjected to hydrolyze to yield simple fermentable sugars. This process, referred to as saccharification, is carried out either by plant materials, barley malt, rye malt or millet malt, or microorganisms, that is aspergillus species, rhizopus species, mucor species rich in starch hydrolytic enzymes. Now let's know about fermentation process. When subjected to fermentation under ideal conditions, alcohol production ranging from 2 to 16% is usually observed. These alcohol beverages may be appropriately processed and drunk. Example, beers. However, for the production of beverages with high concentration of alcohol, brandy, whiskey, rum, gin, etc., distillation has to be carried out to increase the alcohol content. Brewing is the technical term used for the production of malt beverages. Besides S. cerevisiae, Saccharomyces burgenesis is also used in brewing industry. The finished products in brewing, namely the alcoholic beverages, differ from the other industrial fermented products. This is because besides the alcohol content, several other factors are important in finished beverages. These include flavor, aroma, color, clarity, foam production, and stability and satinity. These factors decide the consumer acceptance and the commercial value of alcoholic beverages. All these beverages can be consumed freshly produced when freshly produced. However, they are normally kept for storing or aging before consumption. Aging certainly improved flavors, aroma, etc. The commerce Now let's know about beer. Beer is a undistilled alcoholic beverage with alcohol content in the range of 4 to 8%. It is produced by fermentation of barley or other cereals by employing the yeast S. cerevisiae or S. culbergenesis. It is often necessary to add starch-rich material, that is wheat, maize, rice, known as adjuncts, to enhance fermentation besides reducing the cost of raw materials. Now let's know about malting. First process is, there are four steps in the production of beer. First is malting. Let's know about the process malting. Malting, that means dried barley are cleaned and soaked in water for a period of 2 days. The excess water is drained and the barley are soaked barley are incubated for 4 to 6 days to germinate. Germination process is associated with the formation of enzymes amylases, that is starch hydrolysing, and proteases, that is protein degrading. Germinated seeds are slowly subjected to increased temperature, that is up to 80 degrees centigrade, so that the germination process is halted but the enzymes retain their activities. Malt is prepared by powdering the seeds. Now second process is massing. The powdered malt is mixed with hot water, that is 55 to 65 degrees centigrade. During the course of massing, the soluble materials from malt and malt adjuncts are extracted. Massing is also associated with degradation of starches to produce dextrins, maltose and glucose, and hydrolysis of proteins to peptides and amino acids. The temperature and pH influence the activities of enzymes. The end of massing, the medium for fermentation, which is referred to as beer old, is developed. This is rich in sugars, amino acids, minerals, and vitamins. Hops are the dried female flowers obtained from hop plant. Addition of hops to oat is often done to provide characteristic flavor, aroma, and stabilizing effect to the beer. Besides providing a mild antibacterial activity. Now let's know about the third process, that is fermentation. The beer oat kept in open or closed bioreactors is inoculated with pure strains of yeast Saccharomyces cerevisiae, in short S. cerevisiae, usually top fermenting strains. The fermentation is carried out at temperature 20 to 28 degrees centigrade for 5 to 10 days. In some countries, the bottom fermenting yeast S. Uvarum is used. For this organism, the ideal temperature for optimal fermentation is 10 to 15 degrees centigrade. Now let's know about maturation. The fermented fluid is transferred to storage tank maintained at temperature 0 to 3 degrees centigrade. During the storage period, cold storage maturation occurs. This process is associated with sedimentation of yeast cells and precipitation of nitrogenous substances, resins, phosphates, etc. This partially mature beer, usually with turbid appearance, is then subjected to chill proofing. Chill proofing basically involves the removal of residual proteins. Beer is turbid due to their presence suspended in the beer by precipitating or by employing proteolytic enzymes. It is advisable to add antioxidants during cold storage maturation to prevent oxidative damage. Carbonation of the beer is usually carried out by injecting carbon dioxide. Carbonation can also be accomplished by adding fermenting yeast, which is less commonly done. The matured and carbonated beer is bottled or canned and then pasteurized at 60 degrees centigrade for about 20 minutes. There is variation in the raw materials used for beer production. For instance, in Africa, it is sorghum beer that are commonly produced. This is in contrast to the barley beer produced in most Western countries. Now, let's know about the wines. Wines are originally Middle East and European drinks, although almost every country now produces them. Large scale production of wines is carried out by using grapes of species Vitis vinifera. Vitis vinifera is the grape's name. Grape juice is a good source for wine production because of its high concentration of sugar and other nutrients, natural acidity and the capability to produce pleasant aroma and flavor. Louis Pasteur, a scientist, used to state wine is the most healthy and most hygienic of beverages. This is some recent scientific evidence also in support of this review, since moderate wine consumption reduces the risk for coronary heart disease. In short, CHD, most coronary heart diseases. Now let's know about the production of wines. Quality of grapes is very important for the production of wines. The grapes are crossed and the juice extracted. This grape juice ready for fermentation process is technically referred to as must. It is a practice to add sulfur dioxide to must to inhibit the growth of non-wine yeast and contaminating bacteria. Sulfur dioxide, which can kill other organisms, can be tolerated by wine fermenting yeast. Sometimes the must may also be subjected to partial or complete sterilization. The must in suitable bioreactors is inoculated with desired strains of the yeast Saccharomyces cerevisiae. Initially, oxygen is bubbled through the fermentation medium to promote good growth of yeast cells, and gradually anaerobic conditions are established. The wine production normally takes a few days. That means 2 to 5 days it will take. The fermentation conditions, that means temperature and time, etc., are actually dependent on the type of wine produced. At the end of the fermentation, wines are transferred to storage tanks and allowed to age, which may take some months or years. Aging of the wines is very important for the development of characteristic flavor and aroma. The alcohol content of wines is in the range of 10 to 16%. Now let's know about the types of wines. There are many different varieties of wine. There are hundreds of different types of wines produced in different parts of the world. Let's know about first is red wine. The red color of this wine is due to the color extracted from the black grape skins. Red wines are commonly drunk by many people in the West along with lunch and dinner. Second is white wine. White wine means black grapes after removal of skins or white grapes are used for the production of white wine. Next is third is rose wine. This can be produced with the limited contact of the skins of grapes during fermentation. Fourth is dry wine. It contains relatively higher alcohol content and produced when sugars are completely fermented. Fifth is sweet wine. This is sweet to taste since it contains some residual sugars after fermentation. Sixth is fortified wines. The normal concentration of alcohol in wines is less than 16%. The wines can be fortified with addition of alcohol to the desired concentration. Some examples of fortified wines are sherry, port, and vermouth. Now let's know about the other microbial derived food products. There are a large number of food products or their ingredients produced by microorganisms. For full now, let's know about the points, that is single cell proteins, mushroom, vitamins, amino acids, organic acids, polysaccharides, vinegars. Now let's know about the enzymes in food and beverage industries. Food and beverage biotechnology is very closely linked with the use of enzymes. While a majority of enzymes are microbial in nature, some other sources of enzymes can also be used. Now let's know about proteases in food industry. Proteases are used for tenderization of meat, which flavors the meat besides easy digestion. Proteases are employed for removal of meal attached to bones in the form of a slurry. This can be used for the preparation of soups and canned meats. Partial hydrolysis of certain vegetable proteins also increases their flavor. Example: protein soybean, protein gluten of dough in preparing bread. Now let's know about lactase in dairy industries. Lactase in dairy industries. Milk and whey contain about 4 to 5% lactose. Lactose intolerance due to a defect in the intestinal enzyme lactase is very common in humans. It is estimated that half of the world's population suffers from lactose intolerance. These individuals, when consume milk and milk proteins containing lactose, suffer from diarrhea and flatulence. Obviously, lactose intolerant persons require milk products with a low lactose concentration. The enzyme lactase cleaves lactose to glucose and galactose, which can be easily assimilated by the body. The lactose enzyme may be obtained from microbial sources. Example: Cluveromyces fragile and Aspergillus niger. Sometimes immobilized lactase can also be used to degrade lactose. For the preparation of flavored and condensed milk and ice cream, lactase is widely used. Now let's know about enzymes in the preparation of fruit juice. The turbidity and cloudiness commonly seen in fruit juices is due to the presence of pectins and cell debris. Enzyme preparation containing pectin esterase galactouronase, pectin ligase and hemicellulase is added to the fruit pulp for the removal of pectins and other turbid causing materials. Removal of glucose. Let's know about the removal of glucose and oxygen from foodstuff. The presence of glucose and oxygen in the foods is often associated with certain amount of damage. Their removal enhances the storability of foods. Glucose from the foodstuff can be removed by the combined action of glucose oxidase and glucose oxidase and catalases. Then let's know about the reaction. Glucose plus oxygen in presence of glucose oxidase gives rise to gluconic acid plus H2O2. Next reaction is 2H2O2 in presence of catalase gives rise to 2H2O plus O2. The egg white used in baking industry is made free from glucose by the enzymatic treatment. This treatment. Okay. This pair of enzymes, that is glucose oxidase and catalase, is also useful for the removal of oxygen present in the head space of bottled and canned foods and drinks. Now let's know about diagnostics in food biotechnology. It is essential to ensure that the foods produced by biotechnological processes are free from disease causing organisms and toxins. Then only the foods are suitable for human consumption. The most common pathogens present in foods are Salmonella species, Campylobacter species, and Listeria species. The food toxins are derived from bacterial and fungal sources. Several novel and innovative techniques have been developed in recent years for testing the safety of foods. For instance, the presence of salmonella in foods can be detected by an immunoassay on the same day. This is in contrast to the traditional method that may takes 5 to 7 days. In fact, kits are available for the detection of concentrations of several undesirable compounds in the foods. Example, endotoxins, mycotoxins, antibiotics, and hormones. Now, let's know about food biotechnology, that is public acceptance. In general, public have a negative attitude towards the biotechnology based foods, particularly involving genetic manipulations. For this reason, the application of genetic engineering techniques for food biotechnology is rather slow. Several factors influence the public acceptance. These include the type of biotechnology adopted, the regulatory procedures, economics of the people, and consumer acceptance. Finally finished. Any questions regarding this, you can ask me in the comment box, and can subscribe my channel to know more updates. Thank you.